Hello, my dear students. So, in the previous class, we discussed about other brain syndromes and how we can able to get rid of with the help of stem cell therapy. And today's class is about spinal cord injury and and stem cell therapy. Okay, so let's begin. So, as mentioned, approximately 2 million people worldwide are affected with the spinal cord injuries. One of the most famous was this Christopher Reeve. You maybe see this fellow in Superman film. He is the hero of Superman film. This Christopher Reeve was paralyzed when he was thrown from the horse during a competition. And this review not only set a foundation for stem cell therapy, is a powerful advocate for stem cell research. The major studies cited previously has demonstrated that a new nerve cell can be encouraged to grow and integrate with host brain. There may be a possibility of generation of new nerve cells. Okay, and certain scientists are doubtful that they whether they could reconnect a damaged spinal cord or not. Okay. So it was also realized that most than just nerve cells was required after a damage. This is called syrinx. It's a place where the fluid will be filled in the spinal cord. In the spinal cord, there is a place where fluid or attached. It is called as a syrinx, and it's formed, and the nerves at the injury site became demyelinated. So this oligodendrocytes are also needed to infiltrate and insulate the nerves. So thus, that is begin when the stem cell researcher begin to overcome or replace the damaged nerve cell. So in '99, Jonas and their colleagues and their team they reported that ependymal cells responding to the site of spinal cord injury are actually stem cell. And after that, however, instead of developing into replacement neuron, they differentiated chiefly into astrocytes and which are not functionally able to reconnect the neural pathway. After in '99, Jonas McDonnell at Washington University School, they reported that a positive result using embryonic stem cell on mice who in quarter were paralyzed from a spinal cord injury. Their back foot they it is paralyzed in a spinal injury. Among other things, this McDonnell wanted to see that whether the success, how he would get by waiting for a period before injecting the embryonic stem cell. So 9 days after the injury, the mice were injected with a neuronal precursor that was being derived from embryonic stem cell, and then it was treated with the retinoic acid. And the study was being carried out. So here you can see the study. It's an image. Some eyes. It's having the spinal cord has been index was paralyzed and injected with the into the spinal cord. Stem cell should be injected. You can see that, and there is a recovery of. It will be migrated. The cells will be stem cells will be migrated to the injured area, and injured cells migrate to the injury and wrap around the injured spinal cord. Okay, so there may be hind legs gain function. After that treatment, they found out their hind leg getting a function. So that was an excellent study which was carried out by this McDonald. He injected approximately 1 million cells into the larynx region, and that have formed around the site of injury. The mice had been given cyclosporine to inhibit immune response and to prevent rejection of the foreign tissue. So usually it may be given immune to avoid the immunological response when the foreign body injected. They'll here the McDonald give cyclosporine to inhibit the immune response. And after a few weeks later, some of the mice were killed, or the mice were sacrificed, and their spinal cords were scanned for a new cell. So they found out some of the cells were died and several have grown projection up to 1 centimeter long. Okay, they found out there is a projection and the newly sprouted axons were clearly visible. Okay, so after a month, several of the mice have recovered some of their mobility and coordination. Importantly, none of the mice was developed a tumor, so they don't affected by tumor, but so they found out there is a growth of axons and the newly producing cells. And then in December 2001, Kano at this Kyoto University, Japan, they announced they restored mobility to marmosets. Marmosets is a type of small monkey. Okay, marmosets is a type of monkey whose arms were paralyzed due to the spinal cord injury. Okay, so all the marmosets showed improvement after the transplant and one regained almost full use of its arm. So here you can see They almost showed improvement after the transplant. One regained almost full can use their arms. So this marked the full first successful repairing spinal injury in primitive with the stem cell. Okay. Then Cano used human fetal stem cell, which he was able to amplify to over a million cells suitable for the therapy in a small animals like a marmoset. They found an setup, or they found a base and foundation for this therapy, spinal cord injury. Then in May 2005, Hans and their teamwork, they reported that using a human embryonic stem cell to treat the injured rat, they induced embryonic stem cell to differentiate into early stage oligodendrocytes and then injected them into a rat with a spinal, which is having a spinal cord damage. One group was injected 7 days after the damage, and other group was injected after 10 months. They waited, and after 10 months, in both groups, the embryonic stem cell formed functional oligodendrocytes that migrate to the site of the damage. And but although the earlier groups have showed a significant improvement in the mobility compared with that of second group. So while comparing the 7 days, if their injury is within 7 days, you can able to treat with the help of the stem cell, because this oligodendrocytes moved faster and they'll migrate to the injured area and they'll maybe rectifying the system. Okay, but the second group doesn't have the system of improvement. And apparently, even though the oligodendrocytes established themselves, they could not penetrate the scar tissue and that have formed almost dying neutrons. So that then the oligodendrocytes that were administered after 7 days were able to re-insulate the injured neurons and to provide them with an environmental condition for healing. And this research which will establish the importance of timely repairing of spinal cord injuries. It also indicated that body's immune response to the spinal cord injury that can be counterproductive. And notably, in all the experiments where embryonic stem cells were induced to form precursor of nerve cell, that is an important thing they found out, is maybe producing the precursor of nerve cell. And this indicated that this embryonic stem cells are plastic enough to become a multiple cell line without producing the tumor cells. Okay, so that's all for today. Hope you will understand about the spinal cord injury and how we can cure the spinal cord injury with the help of stem cell therapy. Okay, so if you have any doubts, you can contact me. Thank you all. See you in the next class.